Hi everyone, today we're going to compare cell division between mitosis and meiosis. So let's start off with interphase. Okay, so during interphase, the DNA still exists as chromatin threads, okay, but I'm just going to draw them as chromosomes just for simplicity's sake. Okay, so the DNA actually will undergo DNA replication. Alright, and homologous chromosomes will now each have two sister chromatids joined at the centromere. Then mitosis will start off with prophase, condense to form chromosomes. During metaphase, the chromosomes now aligned in the equator. Alright, and anaphase, what happens? The sister chromatids split apart, okay, and are pulled to opposite ends of the cell. And then telophase, they start together. And after cytokinesis, what happens is that you get the exact same number of chromosomes in each cell as the start. Okay? Now, it is important that we understand a lot of different terms. So, number of sets of chromosomes, for example. So, whether or not it's haploid or diploid at each stage. Okay? We also need to know about how many chromosomes are there or how many sister chromatids are there. And lastly, we should also compare what is the DNA content. Okay, so if we look at these two cells over here, during interphase, they are diploid. Now, why are they diploid? Because you can see that there's two sets of chromosomes, right? One from the mom and one from the dad. So one set of yellow chromosomes and one set of blue color chromosomes. Okay, so there's a total of 4 chromosomes and 0 sister chromatids. But after DNA replication, there is still 4 chromosomes, but now sister chromatids is 8, and the DNA content has doubled to 2 times. So moving on to prophase, so it's still diploid. There is still 4 chromosomes, but 8 sister chromatids, and it's still 2 times. Same thing for metaphase. Now for anaphase though, it is diploid, but there are 8 chromosomes and no more sister chromatids. Why is this so? Okay, only when they are attached, they are called sister chromatids. The moment the sister chromatids separate, they are now called daughter chromosomes. So in this cell, there are actually 8 chromosomes and 0 sister chromatids. Okay, but the DNA content is still 2 times because it's still the same amount of DNA in the cell. Now let's look at telophase. Okay, it's still diploid. There is 8 chromosomes still, and it remains the same as 2 times DNA in this cell. Okay, if they're asking about nucleus, then it might be different already. If the nuclear envelope reforms in telophase, it means that there are actually 2 nucleus, so each nucleus will have 4 chromosomes instead. After cytokinesis, it is still diploid because you see that there is still the yellow chromosomes, one set, and blue chromosomes, another set. Each cell now has 4 chromosomes, and the DNA content is back to 1 times because it is now split into 2. Okay, so it is back to the same original DNA content as the start. Okay, so that's mitosis. Now we will go on to meiosis. So we'll start off with meiosis 1. Alright. So during interphase, it is very similar to mitosis. Okay, so the exact same thing happens. So if I have 4 chromosomes, it's still 4 chromosomes, but now they have duplicated to form 8 sister chromatids. Then during prophase 1, they all will now break and exchange parts. Okay, so this part is different. So during prophase 1, crossing over happens between non-sister chromatids of homologous chromosomes. And what this does is it increases combinations of alleles, so there is more genetic variation. Okay, so you can see that my chromosomes have now exchanged colors. Now, during metaphase 1, you realize that they line up in pairs at the equator. If I actually compare back to mitosis, during metaphase, it actually lines up singly at the equator. Okay, so that is the second difference, that they line up in pairs instead of lining up singly. And when they line up in pairs, this is what we call independent assortment. okay, where different combinations of maternal and paternal chromosomes are split. So depending on how they line up, you might either get your father or your mother's chromosomes. Now looking at Anaphase 1, okay, the homologous chromosomes actually split apart, or they separate into opposite ends of the cell. Okay, if I compare to mitosis, you realize that it is the sister chromatids that actually split apart. Okay, so during anaphase of mitosis, sister chromatids separate, but for anaphase 1 of meiosis, the homologous chromosomes separate. Now for telophase, okay, so the homologous chromosomes now are at opposite ends of the cell, and after cytokinesis 1, okay, the cell will be split into 2. Now you realize that for this cell, each cell has half the number of chromosomes originally. So now they only have 2 instead of 4, and the cells are genetically different. Okay, they're not the same to each other, neither are they the same to the parent. Okay, so that's the first round of meiosis. So let's talk about the various terms again to compare them. During interphase, both of them are diploid because they both have 2 sets of chromosomes from the parents. Okay, so this is exactly the same as mitosis, so we're not going to zoom into that. During prophase, the 2 sets are still there, right? So it's still diploid. There is still 4 chromosomes, 8 sister chromatids, and double the number of DNA content. For metaphase, the 2 sets are still there, everything is still the same. Okay, and for anaphase, for this particular cell, it is still diploid. Okay, and this has 4 chromosomes still, and the 8 sister chromatids are still intact, right? They didn't split apart, so they're still sister chromatids. Okay, so please take note of this. When I look at telophase, okay, it is still diploid. There are still 4 chromosomes with 8 sister chromatids in total and 2 times. Alright, so this is for this particular cell. If I ask for nucleus, then you have to focus on each end of the cell, okay? After cytokinesis 1, you realize that now the cells are haploid because they only have 1 set of chromosomes, right? They are no longer homologous pairs. So they have half the number of sets of chromosomes, so they're haploid. They have 2 chromosomes, okay? So one of the large one of the smaller one, and there's a total of 4 sister chromatids in each cell. And now the DNA content has been halved to 1 times, okay? So that's for meiosis 1. Now, meiosis 2 will follow very quickly after meiosis 1. And for this, you will realize that there is actually no interphase between meiosis 1 and 2. They will just continue on with prophase 2. Okay, so I'm just going to zoom in on one of these cells, so those with the yellow chromosomes. Okay, but you must know that there's also another cell undergoing the exact same thing. So prophase, nothing much happens, they just condense to form chromosomes. Metaphase 2, now what happens is that they line up singly again. Okay, so meiosis 2 is actually very similar to mitosis. So when they line up singly, then during anaphase 2, the sister chromatids will separate and they are pulled apart. Okay, so now they are daughter chromosomes already. And then during telophase, they are at both ends of the cell. They will start to decondense, the nuclear envelope will start to reform again. And after cytokinesis 2, you will observe this. The cells are still haploid because they still only have 1 set. Okay, and they have half the number of chromosomes as original. And please take note, there's actually a total of 4 cells, but I've only drawn 2. So that is for meiosis 2. Okay, now for prophase 2, it is haploid. Okay, there are 2 chromosomes and 4 sister chromatids. and the DNA content is 1 times. Okay, same thing for metaphase 2. Now, but for anaphase 2, you realize that the chromosomes now, there are 4 daughter chromosomes and they are no longer any sister chromatids because they have separated. Okay, DNA content is still 1 times in this cell. For telophase 2, it's still haploid, there is still 4 chromosomes and 1 times DNA. But after cytokinesis, okay, you realize that the chromosome number is now 2 in each cell, okay, and there are no sister chromatids. And the DNA content is now half, okay, compared to the very original cell. I have half the number of DNA. I also have half the number of chromosomes. Now we have this big picture. I want to focus on the genetic variation. So how does meiosis increase genetic variation? The first step is crossing over in prophase 1. Okay, so if we recall, let's look at prophase 1. Crossing over takes place. So when they break and exchange chromosome parts, what happens is that I increase combinations of alleles. Okay, so there is more variation in the offspring. Second thing that I do is independent assortment at metaphase 1. So let's take a look at metaphase 1. Okay, so depending on the way that they line up, you might get more of the maternal chromosomes or more of your paternal chromosomes. Okay, so you have different combinations of yellow and blue color chromosomes depending on how they line up in pairs. And the last thing to increase genetic variation is random fertilization. Now take note that this is not part of meiosis. Okay, so there's many different sperms, many different eggs, and we don't know which one is going to combine with each. So that is actually the overview of comparison, and I hope that this will help you.